Kia ora. Ka tangi te titi, ka tangi te kākā, ka tangi hoki a hau, ti hei Māori ora. E na mana, e na reo, aora rakatira mā tēnā koutou, tēnā koutou, tēnā koutou katoa. Tēnā ka raro e te maru e nā mana whenua, nā te mamoi, waitaha, kaitāhu, nau mai, hare mai, tauti mai. Ki te whare tūnei, tēnā koe, tū tonu. Ina mate haere hoki atu rā ki te pō. Ko Waiao, no Otopoti ahau, ko Richard Blakey tāko ingoa, he kamahi ahau te whare wānanga o tākou, tēnā koutou, tēnā koutou, tēnā koutou katoa. Kia ora everyone, and welcome to this wonderful celebration of promotion to Professor of Rebecca Campbell, our inaugural professorial lecture. My name is Richard Blakey. I'm Deputy Vice-Chancellor, Research and Enterprise here at the University of Otago. It's my pleasure and privilege to welcome you here tonight and set the scene for the event that we have before us. I bring apologies from the Vice-Chancellor, Professor Murdoch, who is very, very sorry he can't be with us, but also as we're welcoming guests online, know that Professor Murdoch will take the opportunity, if he can, should this be recorded, to catch up with the proceedings. Because this is a very important time for our academic leadership at the university, not only to celebrate the success of our most successful academic staff in reaching that pinnacle promotion of professor, but also just to have time where we can uncouple from the other things that we have in our lives and actually just sit back and enjoy and reflect on the wonderful work that our leading academic staff can engage in. So it is a celebration of achievement, and for that, congratulations, Rebecca, on that achievement. It's also a time to celebrate with our academic community, so welcome to colleagues, members of the Dunedin and wider public, so welcome to members of the public, both here, and I understand as far away as California for your father and stepmum, Jim and Deborah, as far away as the UK for your sister, Liz, but also acknowledging your whānau here, your mum Karen, who I understand is here, your husband Andrew, kids Charlotte and Eli, and some in-laws Jane and Christine, and others who might be here supporting you. Welcome on behalf of the university. It's our pleasure to host you here on this beautiful Dunedin evening. So to those in California, it is 50 degrees here, Fahrenheit, and we have liquid sunshine that is in abundance today. So we are enjoying the day very much being indoors. I'm going to hand over to Professor Campbell to give a formal introduction to you for the lecture, but to set the scene, promotion to the professor at the University of Otago is a big deal, and we put on our gowns to celebrate. We undertake a very rigorous process to assess people who wish to be considered for that promotion, including the use of international referees to benchmark performance against other leading universities. And the criteria are pretty simple, but really, really hard to achieve. That we have three core areas of university activity for academic staff of teaching, research, and service. Service both to the university and our wider academic and non-academic communities. And our academic staff members must achieve a very high and sustained level of achievement in at least two of those areas to warrant promotion. And in having a look at Rebecca's CV, I can see that the level of achievement is exceptionally high in all three. Rebecca is an outstanding and very successful researcher. I will mention this, we don't just use money as a measure of success, but bringing in more than $15 million of extramural funding and leading an HRC program, those are really, really difficult and high levels of achievement. She is internationally recognized for that research. She is a very popular teacher and supervisor of research students, highly sought after, having supervised or being a supervisor for 9 PhDs, 3 MScs and more than 20 honours students. And her university service and service to the discipline is very, very extensive and represented at the highest level. So we're very pleased, and I'm very pleased to be here to warmly congratulate you on behalf of the university for your well-deserved promotion. Rebecca, and we'll hand over to Professor Neil Gemmell to give you a more formal introduction. Nō reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you very much.
Tēnā koutou, tēnā tātou katoa, hei pai ki te ki a katoa. No mai, haere mai. So it's my very great pleasure to greet you and welcome you to Professor Rebecca Campbell's Inaugural Professorial Lecture. Richard's already illustrated, I think, the depth of achievement that Rebecca has and why this professorial promotion was so richly deserved. Rebecca is quite simply an outstanding colleague. She is an accomplished teacher, a fantastic researcher, research mentor of the highest calibre, and actually an exceptional academic contributor and colleague. So congratulations, Rebecca. We're so very, very proud of you. Rebecca started her academic journey some part further from here, where indeed the sunshine is not liquid, it is indeed sunshine. So she started in Cal Poly and completed a BSc there, before moving on to the Oregon Health and Science University, or OHSU for short, where she completed her PhD studies in relatively record time, I'd have to say, for the United States. So again, always looking as somebody who was going to progress and achieve exceptionally. She came to New Zealand almost 20 years ago and commenced as a postdoctoral fellow in the Department of Physiology. And she has stayed there ever since, and I think physiology is extremely lucky to have you as an academic contributor to that department, and we're very, very proud of you also in the school. The only thing that you could have done better was to have joined the Department of Anatomy. Bit of parochialism coming through there, sorry. So following that postdoctoral fellowship, Rebecca moved through as a research fellow and then became a lecturer in the Department of Physiology in 2009. So in a very relatively short period of time, she established herself as a leading academic contributor in the area of neuroendocrinology. Rebecca's achievements as a researcher I'll go through in a little bit of detail, but just to acknowledge that as well as being an exceptional academic, Rebecca currently holds the role of Deputy Director of the Centre for Neuroendocrinology, and she is also the Deputy Head of Department Research for Physiology, and sits on the Maurice Wilkins Centre of Research Excellence Leadership Forum. So these are all additional activities that distinguish somebody as being not just a fantastic teacher and researcher, but also somebody who we can rely on to undertake challenging tasks at times. Previously, she's been the Associate Dean Research for the School of Biomedical Sciences, and I remember working with Rebecca when she started that role, and was amazed at how much energy and enthusiasm and drive she brought to that role. We're very appreciative of your time that you spent working on that to bring the school forward. But it's really, and I guess this is, Richard has already illustrated, that we are recognising three things when we promote to professor. But one of the things that really distinguishes our professors often is their research achievements, which are benchmarked internationally, as Richard has already said. And Rebecca's research actually touches to me quite personally, because it works in an area that interfaces between the brain and reproductive systems. And for those of you who don't know me, I have some interest in reproduction. And really it's about focusing on how androgens, which are male hormones, impact the female brain, or predominantly male hormones, and how that then impacts the development of pathology of a disease called polycystic ovary syndrome. And polycystic ovary syndrome probably affects many of the people who are listening online, and also some in the audience. It's the most common endocrine disorder amongst women. It can affect in some populations up to 1 in 5 women of childbearing age, and it can be quite devastating in terms of its effects. And despite its high prevalence, we know relatively little about its cause, and indeed we have relatively few clues about its treatment. But Rebecca is leading the way in terms of developing ways to better understand this and treat this disorder. Her team has discovered that androgen excess drives changes in the female brain and that they are associated with the development and maintenance of PCOS-like features. And she has focused on novel ways to prevent and reverse this, predominantly working with murine or rodent models. I could go on at length and have been known to, but I will draw it short because I don't want to steal Rebecca's thunder. She's going to give us a wonderful IPL, I'm sure, and I'm going to invite her to the stage to deliver that address. Kia ora.
Thank you so much, Deputy Vice-Chancellor Blakey and my Dean, Neil Campbell, for that really kind and generous introduction. I'm kind of blown away. And to the rest of you, tēnā koutou, tēnā koutou, tēnā koutou katoa. E mihi ana ki ngā mana whenua, no karaponia aho, ko Lyle Campbell toko whanau, ko Rebecca toko ingoa, no reira tēnā koutou katoa. Kia ora. So for those that aren't from here, that are streaming in, I've given you my mihi in Maori. I've greeted you, and I have acknowledged the people of the land, and I've introduced myself. So thank you so much for being part of this. Those of you that have made it out tonight on this really horrible weather night, thank you for braving the weather to be here. It means a great deal to me. And thank you also to those that are streaming in. As Richard kindly mentioned, my dad and my stepmom in the US, my sister in the UK, and friends from several places in the US. Great to have you guys here. And I'm really excited to be able to share with all of you, my friends and my family and my colleagues, a little bit about what I do. And the title of my talk is a little bit of a play on words, because as you'll see through the work that I'm going to talk about, a lot of my work involves harnessing light, generally in the form of lasers and microscopes, to be able to visualize these beautiful cells in the brain, to be able to better understand their structure and infer more about their function. But I'm not just going to talk to you about science. The other goal of my talk is really to honor and acknowledge all of the amazing people that have supported me on my career journey and in my life. This Māori proverb asks, what is the most important thing in the world? Well, let me tell you, he tangata, he tangata, he tangata. It is people, it is people, it is people. And in the spirit of that, I really want to dedicate this lecture to my family, both my family of origin that raised me. I'm really excited to have my mom here tonight. Also the family that I've chosen and inherited, and the family that I'm raising, and also to those who are no longer with us. And I'll tell you a little bit more about some of these people as I go. So just like a good thesis or a good research article starts with some background, I'm going to start with a little bit of background about me, tell you a little bit about my research journey, and throw some science in as well. So I hail from the sunny Southern California coast. I've been a lover of the beach and apparently Wonder Woman from a very early age. And I was very fortunate to be born into a family where I was loved and supported and where learning was always highly valued. So these are my parents. This is my mom, Karen, who's able to be here tonight. My mom's always been my greatest cheerleader and an amazing role model for me. This is a picture of her in the Texas A&M yearbook from where she got her master's degree in history. She taught at the high school and junior college level. And yeah, it's just awesome to have you here, Mom. And this is my dad, Jim. You can see him here in his PhD regalia. So my dad is a retired organic chemistry professor. He likes to tell people, though, that he teaches sex education. He said it's makes him more fun at parties. Nobody really wants to talk to an organic chemist, apparently. From my dad, I've learned a lot about the value of hard work, and he's really modeled a love of lifelong learning. I'm also really grateful to my wonderful stepmom, Deborah, here. Hello to you guys in California. And I've also been lucky to be surrounded by some fantastic siblings and step-siblings. You guys are all amazing. And I just want to particularly make a shout out to my 2 older sisters. You guys really paved the way for me in life in general, but also in science and academia. So my sister Elizabeth, Libby, taught science for many, many years and is now a head of school in the UK. And my sister Pam is a professor of psychology in California. But I didn't always want to be a scientist, even though I went to a high school where they had a very good reputation for a great science program. And I loved my biology teacher, Mr. Jindanian, or Mr. J as we called him. And he was very famous for winning the local science Olympiad. And even though I really loved science, I had nothing to do with winning these competitions because I had many other extracurricular activities. And some of you may not know that I captained our very competitive drill team for many years. So this is mostly what I was doing in high school. And I was also having a wonderful time with this crew of friends who really became my lifelong friends. This is us about 30 years ago, and this is us just last month. But by the time I finished high school, I had figured out that I really loved science, and I liked the idea of helping people. So I ended up moving a few hours up north to become a pre-med major at Cal Poly San Luis Obispo, and then beautiful Central Coast. And again, this time is mostly punctuated for me by the amazing people. These are my flatmates, or housemates, I suppose, as we call them in the US, and these are still my precious friends. But there were many things that happened during these years at Cal Poly which really ultimately led me to where I am today. So the first of those things was that during my pre-med program, we were encouraged to work in clinics or in hospitals, and I ended up working in an obstetrics and gynecology office. And this is where I first learned about polycystic ovary syndrome and the devastating effect that it has on women, and really learned to value women's health and how little we really understand about it. But I also learned that being a physician was probably not for me. So that was the first thing that happened. The second thing that happened was that I had the pleasure of interacting with this inspiring woman. She was a young, scrappy academic who had just left her postdoc at UCSF and come to Cal Poly, and she was so excited about her teaching and research. And I could see in her potentially what I might be able to become if I went down this research and academic path. And it's just reminded me of the value of role models for all of our students. The third thing that happened was that I decided to spend a year abroad, and I ended up more by process of elimination than anything else at Lincoln University in Christchurch. And two really important things happened there. So the first is that I had a chance to do some whole animal physiology, and in particular neuroendocrinology, with Graham Burrell, who was very inspiring, and also happened to be Professor Greg Anderson's PhD supervisor at the time. So I got really excited about the idea of being able to test what's going on in the brain and measure hormones in the periphery. The other really important thing that happened was that I met the love of my life and my future husband, Andrew. So by the time I finished my years at Cal Poly, I knew that I was probably not going to medical school. I was probably going to go to graduate school and check out research, and I was probably going to spend a lot more time in New Zealand. And my family took it relatively well. So being inspired by my time in the clinic, I decided that I wanted to do research on the ovary. And so that took me to Oregon Health and Science University up in beautiful Portland, Oregon. You can see Mount Hood here in the distance. And although I intended to join a lab that studied the ovary, through the course of my first year lab rotations, I had the opportunity to work in some labs that were looking at the neuronal control of the ovary. And I decided that perhaps I should be studying the organ in the body that controls the ovaries. And so I ended up joining the laboratory of Susan Smith, who is a formidable scientist and inspiring mentor. She was the director of the institute that I worked at, and I'm really so thankful for all of her support. And I also had the opportunity to work with Kevin Grove, who was a lab manager at the time, incredibly supportive. I knew nothing, and he essentially taught me everything I know. So my project was about looking at the circuits in the brain that integrate signals about metabolism with the circuits in the brain that signal reproductive function. And specifically, I was investigating how negative energy balance is able to suppress reproductive function. So just to set the stage for this concept, give you the rationale. So we know that individuals that are either underweight or overweight will frequently suffer from infertility. And lifestyle changes that bring those individuals into a healthier weight will move them into a place where fertility is generally restored. So we know that energy balance and fertility are tightly linked, and we know that this is actually happening in the brain. So here's the brain, and I'm going to gently introduce you to the circuits I want to tell you about today. So there's an area at the base of the brain known as the hypothalamus. And in this region of the brain, there are networks of neurons that ultimately control the release of hormones from the brain that then go on to drive patterned secretion of additional hormones from the pituitary gland, which go on to regulate what happens at the ovary. And this includes things like ovulation and also the production of gonadal steroid hormones like estrogens and progesterone and androgens like testosterone. And these hormones have important feedback effects at the level of the brain through this neuronal network that controls hormone output. But this network is not only responding to steroid hormones from the periphery, it's also importantly responding to other types of signals that signal whether it's an appropriate time for reproduction to occur. So this includes things like energy balance, and nutrition, and stress. And at the time that we were looking at this was 25 years ago, how this was actually happening in the brain was really poorly understood. We were just beginning to understand it. And so I'm not going to go into any great detail about the data that I collected in my PhD. I just wanted to highlight this as a time in which I learned how to use antibodies and fluorescent markers and fancy microscopes to be able to identify specific cells in the brain and the receptors that they express and the interconnections between neurons that are passing on information about things like energy balance. So these are just some images that I took with my phone out of my printed thesis because I can't access the floppy disk anymore. Yeah, I should have probably done something about that earlier. But you can see some of these beautiful cells and all of these connections from other cells. These are axons, these long fibers that come off of neurons. And we looked at where receptors were expressed on these cells to identify a little bit of the functional anatomy of energy balance signals that control reproductive function. I was pleased to have some of this work grace the cover of Endocrinology. And I just wanted to show you my summary diagram for my thesis. It's very complicated. I won't walk you through it. But just to say that we were able to set the stage for understanding which neurons in the brain are communicating to other neurons in the brain and able to respond to these signals about energy balance. This is me looking very happy after I've just defended my thesis publicly. And yeah, again, I'm really grateful to Susan and Kevin for all of their support through these years. And I can't leave this period of my life without acknowledging very wonderful people that were my mentors and supporters and friends through our time in Portland. And so at the end of this, of my PhD, or close to the end of my PhD, Andrew had moved to the US and we were quite happily settled there. And we didn't really intend to leave the US, but I received a newspaper clipping. It was actually an article out of the Southland Times that had been sent from Andrew's family, talking about a researcher in Cambridge who was moving to Otago to establish a research center on neuroendocrinology. And I thought, gosh, what a great opportunity to go and do some research I'm really interested in, and also to be back near Andrew's family for a little bit. We thought we'd come back for 3 years, and then we'd go back to the US and get real jobs. We didn't really think we were moving here for life, but that was 20 years ago. And it's because it's so good. So this was Alan Herbison, who was moving from Cambridge, and I was really pleased to be able to join his lab. So this is the early lab. You may recognize some of these young, fresh faces. And he established the Center for Neuroendocrinology, and this is our newly minted group. And I'm really grateful for this research group of really collegial and collaborative colleagues. It's been an amazing group to be a part of. We do a lot of baking, and these are all the individuals I think that had something to do with the US. I think we'd all studied in the US and we were celebrating the 4th of July. So these were some great years as a postdoctoral fellow and a research fellow in the Herbison Laboratory. And the projects that I worked on were really somewhat similar and a bit of an extension of those things that I'd done as a PhD student. The focus was really, though, not on energy balance, but on these gonadal steroid hormones and defining the neuronal circuits through which they were working to regulate reproductive function in the brain. In addition, we were also interested in looking at these neurons, which are the final output neurons of this large neuronal network that regulates reproductive function. These are known as the gonadotropin-releasing hormone neurons, or GnRH neurons. And I was interested in determining how these neurons are poised to receive and integrate all of the different signals that they're receiving from other cells to regulate their function in the varied ways that they need to behave over the life course of reproductive function. And one of the really cool parts about the work that I was able to do was that it involved these new transgenic models and tools that were just coming online at the time. So why do we need transgenic tools? What are these? Well, I'll give you a rationale for why we need these. So I like to tell my students that the cortex and the hippocampus and the cerebellum are the salad bar of the brain. So they're full of cells that are all similar to one another in nice, neat little rows. They're easier to kind of go in and identify and study. They're not really that easy, but easier than the hypothalamus, which is really like the soup of the brain. So the hypothalamus is full of all different types of neurons that are sitting right next to one another. And so it can be really challenging to go in and study a specific cell type in the brain. And the GnRH neurons in particular are quite challenging because they're really scattered throughout this soup of the hypothalamus. And so we've really needed better tools to be able to target and study these neurons. And what I want to do is just walk you through one study in my postdoctoral years that took advantage of some of these transgenic tools and tell you about how that model enabled us to learn a little bit more about the central regulation of fertility. So that model is a relatively simple model. It takes advantage of a gene in jellyfish that expresses a green fluorescent protein. And Alan Herbison had generated a mouse model in which that protein is expressed exclusively in the GnRH neurons. So what that means is that we can look at the brain and we can actually visualize these neurons and their scattered distribution throughout the brain. And then we can do lots of cool things. We can actually patch onto these cells with tiny glass micropipettes. And we can fill them with low molecular weight dyes. We can also record their activity and pull out their intracellular contents to study them. But one of the projects that I was involved in was in collaboration with Song-Kyu Han in the lab. And we were filling these cells and recognizing that the morphology of these neurons, when we filled them with these low molecular weight dyes, was quite different to what we had previously appreciated. And it really began to change the way that we think about how these cells are regulated. So if you just look at this higher magnification cell, you can see that when we fill these, all of a sudden we can appreciate all of these new features. So one thing I'll point out are just all of these tiny little protrusions. These are known as spines. And in most neurons, these spines are sites of synaptic input. So this gave us a way to measure the amount of input that was coming to these cells and take a look at the different types of inputs that these cells were receiving. The other thing that we noticed was that the input zone of these cells, which is known as the dendrite, was incredibly long, much longer than we had previously appreciated. And even at the very distal ends of these dendrites, these input zones, we were seeing all of these spiny protrusions, suggesting that these cells were receiving inputs at parts of the neuron we had never actually even been able to see before. And so several studies of this nature allowed us to understand a little bit more about how GnRH neurons function in the short, medium, and long term. And so if you just look at these lines over here, these different patterns of lines are indicating the activity and the hormonal output of these neurons on an hourly basis. So we see these pulses of secretion. On a daily basis, so we see changes in the output, for example, over the female estrus cycle, and also over the lifespan. So we see activity elevated around birth, and then again with pubertal onset. And so I won't go into the details again, but several of these studies helped us to appreciate how GnRH neurons do this, how they behave so differently. So one of the studies involved looking at, there we go, yes. Looking at these GnRH neurons and how they were associated with one another. One of the big questions we've had is how this scattered population is synchronized and can behave together as a unit to generate these pulses of activity. And by looking at these GnRH neurons and how they relate to one another, we were able to posit hypotheses about how these neurons might be behaving together to secrete these pulses, which are required for reproductive function. We also found by looking at the shape or the morphology of these neurons that there were sometimes dramatic changes in the shape of these cells at times when the activity of these neurons was increased. So in the female cycle at the time of ovulation and also with pubertal onset, we see that the morphology of these neurons is changing, suggesting that there's plasticity or changes in the innervation to these cells. Now, I'm really proud about several of the studies that I was able to do in the Herbison lab. This morphological work was also aligned with some work we were doing with viruses to map the circuits that regulate GnRH neurons. But by far the experimental outcome that I'm most proud of during my postdoc and my research fellow years are these guys. So during these years, we had Charlotte and Eli, who are here in the audience. Best experimental outcome ever. Still collecting data. It's kind of longitudinal. But yeah, they're remarkable people, and yeah, you guys are the best thing I ever did. Now, having kids when you're aspiring to be an academic is a little bit challenging, but I had amazing support, both from the University of Otago and my department and my supervisor, but also at home. Andrew's an amazing father and partner. And they say it takes a village to raise a family, and so we have an amazing village. So I'm really super grateful too to my mom, Karen, and the wonderful aunties to my children, Jane Campbell and Christine Donnelly, who are also here. I honestly couldn't do what I've done over the last couple of decades without your love and support. So thank you very much. And I just want to highlight here, I love this picture. This is my mom carrying a very colicky Charlotte while I'm presenting at the Society for Neuroscience conference. So there was some real dedication by our village. So fast forward a few years, and in 2009, I was thrilled to be appointed as a lecturer in the Department of Physiology, which has been a wonderful place to work. And I'm very grateful for my early lab. These individuals really established all of the work that we've been able to do over the last several years. And I want to make a special shout out to Mel Prescott, who I think is in the audience. So Mel has been with me since the beginning. She's an exceptional lab manager. And honestly, I don't think my lab would run without her. So thank you, Mel, for everything you've done. We all love you, and you can never leave. So this is a slide from my 2009 job talk, and so it gives you an idea of some of the things that our lab was focused on. But I just want to spend the rest of the time talking about this project, which was really aspirational at the time, which was to understand what's happening in the brain in this common endocrine disorder, polycystic ovary syndrome, or PCOS. So what is PCOS? So a PCOS diagnosis involves 2 or 3 of these cardinal features. So they include androgen excess or elevated male-like hormones or male-typical hormones like testosterone, menstrual irregularities, and a polycystic appearance of the ovary. So it's quite a heterogeneous disorder. It's also associated with several comorbidities. So it's associated with a metabolic syndrome, including obesity and insulin resistance. And women with PCOS are at a higher risk for developing anxiety and depression, some cancers, and also cardiovascular disease. And it's also really common. So as Neil said, it's estimated that probably more than 1 in 10 women in their reproductive years will suffer from PCOS. And it's currently the most common cause of infertility associated with anovulation. And beyond just the pain and suffering of the women who are coping with PCOS, it has a huge healthcare cost. So the healthcare cost in the USA alone is exorbitant, and we don't really know what the healthcare costs are here in New Zealand. All of the treatment for PCOS is symptom-based, and that's because we understand so little about it, and there's currently no cure. So this was a space that I was really interested in getting into and trying to get at what causes PCOS and how we can reverse it. So why do we think the brain is involved? Well, hopefully you've stayed with me by now, and this is a comfortable concept that the brain and the ovary have this two-way communication. So signals from the brain are controlling what happens at the ovary, and the ovary has signals in the form of hormones that signal back to the brain to keep this balance and homeostasis. Well, we know in polycystic ovary syndrome that this communication is broken down. So the brain appears to be insensitive to the signals that are coming from the ovary. And because of that, you then have hyperactive signals coming from the brain back to the ovary. And we know that this drives many of the pathological features that we see at the ovary in PCOS patients. So how do we know this? How do we know there's this communication breakdown? Well, we can measure evidence of the hormones resulting from this axis in the peripheral blood. So we can look at luteinizing hormone, which you can see here. And we see that its rate of secretion and its level of secretion is dramatically increased in patients with PCOS compared to healthy controls. So this is reflecting increased GnRH secretion from the brain. And we know that this disordered pattern of hormones is driving many of the downstream features. So this impairs ovulation, it causes an increase in androgen production, and androgens are associated with all of these overt and distressing symptoms of PCOS, which include excess hair growth, male pattern baldness, and acne. What we also know about PCOS is that it takes a lot higher gonadal steroid hormone levels of estrogen and progesterone to slow down this hyperactive secretion. And that tells us that the brain has become insensitive to this steroid hormone feedback that would ordinarily slow down this secretion. So that really points to the brain as a central site for pathology in PCOS. And this is something that I thought, given what I'd learned in my PhD and my postdoc, that I could apply to this problem. Now, I'm most interested in women and clinical PCOS, but to study this question in women currently is impossible. We just don't have the tools to look at circuit level problems in the human brain. And so we're dependent on animal models, but animal models have a couple of advantages. So the first is that the hypothalamus that we're interested in is highly conserved across mammalian species. So we can learn what's going on in the hypothalamus of a mouse, and we can translate that back to the clinic. And secondly, using these murine models allows us to take advantage of all of these transgenic tools that allow us to begin to pick apart what's happening on the circuit level. And before I tell you about the work that we've done in this space, I think it's really important to just take a moment and honor and thank those that have come before me in this space. So these are all of the people whose work really set the stage for us to do what we do. So these two clinicians described this hormone output that suggests that the brain is involved. And all of these basic researchers have begun to identify how androgens are acting in the female brain. And I'm really proud at this point in my career to call actually all of these people co-authors and colleagues and friends. So the first thing we needed to do in the lab was to establish a model. And establishing a model can be difficult if you don't actually know what causes a disease. But we do know that excess androgen exposure early in development is strongly linked with PCOS development with puberty. And that's represented here by this red line. So we can take advantage of this knowledge, and we can model animals that express PCOS features by exposing them to elevated androgens early in development. If we expose pregnant dams to high androgens late in gestation, we see that their female offspring develop many of these features. And so we've gone about characterizing this model. They have androgen excess, they have modified morphology of their ovaries, and they are acyclic. So they don't have normal cycles. And importantly, this increased hormone output from the brain is also evident in this model. So obviously different scale, but we see in our preclinical model this hyperactive secretion of hormone. So what I want to do is give you the takeaway message first, and then using as few graphs as possible, hopefully provide you with some evidence to support these claims. So what I'm going to tell you is that prenatal androgen excess programs changes in the brain, programs changes in the wiring to these GnRH neurons, and in particular, changes wiring coming from GABA neurons that signal with GABA. I'll show you that if we activate this circuit that we've identified, we can elicit a PCOS-like phenotype, suggesting that the pathology in this circuit is sufficient to drive PCOS. And I'll also show you that these circuit abnormalities are programmed early, likely associated with changes in microglia pruning. I'll tell you what microglia are. And that importantly, this circuit remains plastic, and we can actually reverse these changes with anti-androgen treatment. Okay, so Alicia Moore was my first PhD student. Wonderful student. She has her own group in the US now. And I was going to say, I learned how to be a supervisor with Alicia. Thank you, Alicia. No, she was amazing. And the thing that she went about doing in the lab was trying to identify the inputs to the GnRH neurons, to map those inputs and see if they were different in our model of PCOS. And so one way to do this is to label for presynaptic terminals. And so this is a GnRH neuron, and all of these little red dots are indicating these presynaptic terminals. And so she could map them along the length of this input zone to the cell. And what she found was that there was a significant increase in the amount of input coming from these neurons that signal with GABA. So what is GABA? This is a bit of Neuroscience 101. So we'll start on this side, and then I'll go to this side. So GABA is a neurotransmitter. It's secreted from nerve terminals in the brain, crosses this little synaptic cleft, and it binds to a variety of receptors. One of these receptors is a channel that allows ions to enter the cell, and when it does that, it's generally inhibitory to cells. But the GnRH neurons are special. They have high intracellular chloride, and so it means that when GABA signals to these cells, it can be excitatory. So we'd found this increase in inputs from a neuron that can excite the GnRH neurons in a model in which we have hyperactive secretion of hormone from these cells. Now this data aligned with data that was coming out of other groups and also with some evidence in the clinic. So we'd known for quite a while that GABA-acting anti-epileptic drugs could drive a PCOS-like phenotype or PCOS features in otherwise reproductively healthy women. And more recently, it's been shown that PCOS patients have a higher level of GABA in their cerebrospinal fluid. I'll just pass that one. So identifying this increase in inputs to the GnRH neurons coming from GABA neurons was kind of exciting, but we needed to find out where these neurons were coming from. So another thing that Alicia did was to take advantage of transgenic tools and viruses that love to pass through neurons, and she looked at different populations of GABA neurons in the brain and followed their long axons to see which of those were connected with the GnRH neurons, and you can see those here labeled as red. And she found that when she targeted the population of GABA neurons in an area of the brain known as the arcuate nucleus, which is an area we know is important for steroid hormone feedback, and she followed the long axons of these cells, she found that many of these axons were contacting the GnRH neurons. And if she looked in our animals that model PCOS, the amount of contact from these neurons was nearly doubled. So we had identified a specific circuit that appears to have enhanced innervation to the GnRH neurons. So this gave us an anatomical framework for a potential mechanism of hyperactive regulation of these neurons in a PCOS-like state. So then we wanted to ask several other questions. We wanted to know what happens when we activate this circuit. Is it actually a functional circuit? If we activate it, do we get hormone secretion? And secondly, is this thing that we're seeing in the brain, this miswiring, is it actually responsible for driving PCOS-like features? So we wanted to chronically activate this population and see if we could recapitulate PCOS features developing. And so the way that we did this was using a couple of novel transgenic tools. One of these is known as optogenetics. This allows us to turn neurons on acutely. And what this does is it uses light to turn specific neurons on and off. To address whether chronic elevation of this population drives PCOS features, we use something known as chemogenetics. This uses designer receptors and designer drugs that allow us to dial up or dial down the activity of specific populations. And so this first question was addressed by Dr. Mauro Silva, who was another PhD student in my lab. He's since done a postdoc in France, and he now has a research position at Harvard. So he took advantage of these tools known as channel rhodopsins. So these are blue light sensitive sodium channels, which means that if you shine a specific wavelength of light onto them, they open the channel, sodium ions come in, and this causes excitation. And so he was able to express these receptors specifically in this population of GABA neurons. and then insert optical fibers that allowed him to flash this blue light onto the terminals of these cells, excite them, and get them to release neurotransmitter. And in the meantime, he measured hormone secretion from the periphery. So he flashes blue light, he's measuring hormones, and what he found was that when he turned the blue light on, he was able to drive secretion of luteinizing hormone in the periphery, and this is a proxy for GnRH secretion coming from the brain. So this suggested that we had a functional circuit. This next question was addressed by Dr. Elodie Desrosiers, who was a wonderful postdoc and research fellow in the lab. She had a Hercus fellowship, and she's recently moved back to France to lead her own group. So she took advantage of these other tools known as DREADS, which stands for Designer Receptors Exclusively Activated by Designer Drugs. And so these receptors could be selectively expressed in our population of GABA neurons again. And then by delivering a designer drug that only acts on this receptor, we were able to dial up the activity of these neurons over time. And what she asked was whether in reproductively fit animals, increasing the activity of this population over time with the delivery of this designer drug in the water would elicit the development of these PCOS-like features. And what she found, without showing you any graphs at all, was that this did recapitulate many of the features of PCOS. So we saw that these animals had increased testosterone after they'd been on this drug. Their estrocyclicity was impaired. They had evidence for reduced ovulation and a trend toward hyperactivity of this reproductive axis. So these data suggested that we'd identified a functional circuit then that, when activated chronically, was able to drive PCOS, suggesting that this impairment was sufficient to drive PCOS pathology. So that raised several other questions. We wanted to know, okay, well, how does this happen? When does this circuit abnormality develop? And really importantly for therapeutics, can it be reversed? So these questions were addressed by Mauro and also another really talented, recently completed PhD student in the lab, Dr. Aisha Sati. And what they did was to look at this circuit and what might be causing these changes over the life course of PCOS development. And what Mauro found was that at about 3 weeks of age in our model, just prior to pubertal onset and this increase in androgens that we see, the GABA input to the GnRH neurons is already elevated, and elevated to levels that we see in the adults. So this suggests that these wiring changes are programmed early by this excess androgen exposure. And what Aisha did through her PhD was to identify that probably what was happening was involving these funny-looking cells here. These are microglia. They are the innate immune cells of the brain, and they're also really important in pruning back synapses in the developing brain. So they eat up all of the excess synapses that are not required to give an adult appropriate complement of inputs to any given cell. So she found, by looking at a time point just prior to when we see this enhanced GABA input to the GnRH neurons, that something was going on. And so again, we used transgenic animals to do this. So these animals have green fluorescent protein in the microglia, this beautiful looking cell here. And what Aisha did was that she labeled for phagocytic vesicles. So these are essentially the trash compactor of the microglia. So they eat up synapses that are not required, and they put the debris into these vesicles. And she also looked for evidence of debris from GABA terminals specifically. And so hopefully, if I do this, you can see that a little bit more closely. So if we turn around these beautiful images that Aisha, let's just go back one more time. There we go. We'll just move right along. We have more moving microglia. Hopefully you can appreciate those trash compactors of the cell here in blue And the pink here is the debris of GABA terminals that have been phagocytosed or eaten by these microglia. So she was able to measure this in our controls and our PCOS-like animals. I'm not sure what happened to my little movie here. But what she found was that there was a reduced volume of GABA terminal debris in the phagocytic vesicles of microglia in our brains from our PCOS-like mice, suggesting that there's a reduction in the refinement of these GABA synapses onto the GnRH neurons, and that might underpin this program change in enhanced innervation. But excitingly, we find that we can reverse these circuit changes even though they've been programmed early. So this is Mauro's work, and he found that anti-androgen treatment for several weeks was able to restore a completely normal level of GABA input to these GnRH neurons. And this was associated with a restoration of cycling, an improved morphology of the ovary, and evidence for increased ovulation in these animals. And so, of course, we were very interested in taking what we had learned about this anti-androgen therapy that was able to reduce this pathological circuit and ask what's happening in women. And the ability to do this was really born out of the time that I spent on my research and study leave, my RSL. And I just want to take a moment to thank the university, thank my department for my research and study leave. Such an important time I really value, and it's paid back in dividends so much for all of the research we've been doing in the lab. So picked up the whole family, and for 6 months we hit the road, and we did a whole lot of things, including a lab project at the Salk Institute in Ed Callaway's lab, which is a whole additional talk, which I won't give you tonight. But during that time, importantly, I got to spend some time with this lovely woman, Inger Sundström-Parama, at the University of Uppsala in Sweden. She's a research clinician in obstetrics and gynecology. And while I was there, we looked at some of her data and we wrote a paper. And we also thought up some ways in which we could start to probe the clinical data for how anti-androgen therapy may impact reproductive function in women with PCOS. And so that study became part of a Health Research Council funded program that I lead alongside Greg Anderson and Christy Gisoni and all these lovely people. And what we did, and this involved Elodie here, who's in the back, along with Inger and her colleague, Evangelia Elenis, was to look at retrospective data, retrospective clinical data, to ask, to look at the association of anti-androgens and fertility rate. And essentially we found two really important things. And I'll just tell you first that what we did. So we used the amazing patient data that's available in Sweden. So we had records from over 200,000 women, and we could ask about their PCOS diagnosis, what drugs they were taking. We could look at their fertility rate. We could look at all this amazing data. And what we found was that women that were hyperandrogenic had the lowest fertility rate. So these high androgens are really associated with impaired fertility. And importantly, we found that if these women were taking anti-androgen treatment early, they had a better fertility rate than those who were initiating anti-androgen treatment later. And so what this tells us is that early diagnosis and treatment could be really key in the prevention of fertility-related morbidity in women with PCOS. And this is really important because anti-androgen therapies had never been associated with any restoration of reproductive function in women with PCOS. And so in the last part, hopefully you can take away these messages, that we've been able to use preclinical models to make discoveries about modified neuronal wiring that might underpin PCOS pathology. And although these programs appear to be These changes appear to be programmed. We can reverse them with anti-androgen drugs. And retrospective data suggests that early use of these anti-androgens correlates with greater fertility rates. So now I get to congratulate you for making it through the heavy science part of my talk. Well done. And I hope that you will just bear with me for a few more minutes while I take a moment to just thank a whole lot of really fabulous people. Oh, before I do that, gotten ahead of myself, let me tell you what I'm doing next. So where have these studies led to? What we're doing now in the lab, what we're funded to do at the moment, given what we know now about anti-androgen therapy, restoring fertility, we're really interested in what the key androgen targets are in the brain. So we're using some really cool transgenic tools to take androgen receptors out of specific cells and see how this impacts reproductive function. Okay, now I'm going to show you some pictures. Okay, so none of the work that I've just shared with you would be possible without these incredible people. So the members of my laboratory team, I'm just going to cycle through some pictures of their beautiful faces and the fun things that we've done. I'm so very grateful to all of you. It's really what gets me up in the morning, is being able to work with these amazing people. I hope that you've learned as much from me as I've learned from you. It's just such a pleasure to work with all of these guys. So thank you for all of your hard work and dedication. I also want to thank my many collaborators, mentors, and just general career champions. I've received so much support and collegiality from so many people, and I think that's really what makes the research community run. So I'm just so very, very grateful for that. This is not exhaustive, so if you're not here, I love you too. And of course, it takes many people, really, to keep me sane and grounded. So I just wanted to take a moment to thank the crew at Musselburgh Baptist. You guys are just amazing. Thank you for coming out. And yeah, just a really loving and gracious place for me and my family. I need to thank my running crew. Thanks for coming out, guys. So thanks for running ridiculous distances with me, and coffee and therapy and all the other things that we do on a weekly basis. You guys are amazing. Thanks for coming out. And I also want to thank our Timaru crew. So these are our wonderful family friends who we spend a lot of our school holidays with. Really love you guys. Thanks for all the support over all of the years, and thanks for coming down in this horrible weather. And of course, Team Campbell. Thanks, guys. Huge thank you to my husband, who, Andrew, who's just an amazing support. Always encouraged my ambition and been a really compassionate ear for all of the things I have to talk about. And you've always believed in me, even when I haven't believed in me, so thank you. And of course, my marvelous kids. Thanks, guys. Thanks for putting up with a really busy and stressed out mom sometimes, and for putting up with me talking about puberty at the dinner table. These guys are so talented, and I'm just so excited to watch you guys grow. Love you guys. And finally, these are names of all of those wonderful pictures that you've just seen. And I also want to take a moment to thank the Department of Physiology, so my amazing colleagues, half of whom have COVID at the moment. So sorry, guys, if you're watching from home, get well soon. And all of the amazing professional staff. You guys make what we do possible. So thank you very much for your patience with us. I want to thank all of the research groups that I'm involved with, all really inspiring people. The Omni staff make it possible for me to use all of these tools and microscopes, so thank you to them. And of course, the BRF staff that look after our tiny little patients and make what we do possible on a daily basis. Thank you so much. And of course, we couldn't do what we do without the trust of the community and the government that support us to do this work. So we're really grateful for that funding. And that just leaves it for me to thank you for being here and listening to me for this whole time, and also for taking time out to celebrate this with me today. It means a lot. stay here? Do I go there now? Okay.
Tēnā koutou, ko Fiona McDonald, taku ingoa, ko au te tumuaki o te tari o mātai whai awararo. So my name's Fiona McDonald and I'm Head of Department of Physiology and fortunate enough to have been Rebecca's HOD for the last nearly 5 years. And it's my pleasure to give a formal thank you to Rebecca. What a wonderful talk. And many, many congratulations on your promotion. And as I wrote the promotion application, you know, it was very so well deserved. So congratulations again. And I want to comment on a few of the things about your talk that have really showcased your strengths and things that we've seen along your journey. The passion that you have for neurophysiology, for those connections, you know, that's very obvious and it's clear now from your student days that's been there for a long time. And also you have this theme, you tried, you know, the ovary was always there, but you had to go via the brain to get that connection between the brain and reproductive function and fertility. And I remember, because I was on your interview panel in 2009 when we employed you as a lecturer, and you came out of nowhere with this, well, I'm going to work on PCOS, and we all looked around and thought, what is this? You're a neuroscientist. But it's so wonderful to see that journey, that story that has come through, and you've made some amazing contributions in that field. You have an international community now that you're part of, and you know the future is bright in terms of making a difference and moving from the preclinical to clinical, which we're really excited to see. One key theme, I think, of Rebecca's work is her getting ahead of the game on the tools, and whether it's transgenics, the dreads, the imaging, you're amazing, you know, all those years in the PhD lab, I'm sure, getting the imaging and the working out the knobs on the confocal, you know, I think that's been really pivotal for you to get to where you are. We all know that putting in kind of those fresh, exciting techniques into your grant applications really make a difference. So I think that's something that you've done incredibly well and used your collaborations and connections as, to enhance that. And then I think as well, which has been kind of touched on already, is that you have made yourself big in the international arena. And that's very obvious from, you know, those PCOS gurus who, you know, you collaborate with and have published with. That's very, very impressive. We're in Dunedin, you know, they're all around the world. And through your work, you've become part of that community. And as I think Neil said, you're involved in governing bodies for PCOS, for example, that International PCOS Society, helping to organise conferences that are the pinnacle international conferences, and various editorial duties, reading chapters today. So working with allowing scientists to publish their work in very good journals. But it's not only that, your strength as a researcher, as kind of a colleague and a superb postgrad supervisor has meant that Rebecca's been tapped on the shoulder or put herself forward for these many roles that support research in the department, as my Deputy HOD Research, as Chair of our Research Committee and member of our Research Committee, the Associate Dean Research for the school. You were postgraduate coordinator for a couple of years. You were 400 level convener for 4 years. You've made an enormous contribution to the research culture in physiology. So, and that's really come through again through your journey, I guess, and sharing with others and supporting others. So that's really appreciated. And you have been recognised for this through several awards, Emerging Researcher Award in the school level and Distinguished Researcher Award just a couple of years ago in the school. So you've brought, you know, the research, but it moves out into all those the teaching, the service, and supporting an excellent culture of research. And finally, I think Rebecca has shown us today her skills at scientific communication. You all know how the brain controls fertility and reproduction now. So thank you so much for that very clear and illuminating talk. So I think I'll just stop there and say please join me again in thanking Rebecca and for a wonderful talk. And I have the pleasure of giving you a gift on behalf of the university to mark this occasion. So enjoy that. Thanks.
Thanks, everyone. Thanks, Fiona.